Our work deals with understanding the phenotypic and epigenomic impact of SARS-CoV-2 nucleocapsid protein, or the N protein, in breast cancer. So the idea here is that once SARS-CoV-2 enters our cells, some of the proteins, and specifically the N protein, localizes to the cell nucleus, and there it would trigger changes in the host genome, impacting the transcription and pathology of the cell. So this is some work done from a previous lab last year, Dr. Nevin Krogan's lab at UCSF. And this is showing you all the different proteins found in the SARS-CoV-2 genome. And down here, you can see some of these proteins they found interact with a number of interesting chromatin proteins. Further, the expression of ACE2 in breast cancer can vary based on the subtype. As you can see here, for example, the breast invasive ductal carcinoma has higher expression of ACE2 compared to the others. So this is our hypothesis that this N protein would localize to the nucleus of the cell, and the effects of the protein is also going to vary based on the breast cancer subtype if it has differential expression of ACE2. So some of the students in the lab tested a few cell lines that belong to these specific subtypes of breast cancer. For example, T47D is luminal, expresses luminal markers, T31 is mesenchymal, or highly basal in origin, and then these do not express these markers. We also have spent some time making a lentivirus expression vector to express N protein in these cells. And when we look at this via immunofluorescence, blue represents the nuclei, the red is the N protein. When we zoom in on some of these cells, we can see expression is both nuclear as well as cytoplasmic. So this is very exciting. And we are looking forward to using these cells to test for RNA expression, transcriptomics, in other words, as well as chromatin accessibility to see how expression of N protein affects the epigenome. And thanks to the DaCCoTA pilot program for supporting this work, as well as the NIH Cobraid epigenetics group and the REMS program for medical students. Thank you for listening.